Have you ever wondered how your muscle actually move, whether you are sprinting, lifting weight, or even just blinking? There is one protein at the heart of all, myosin. This molecular powerhouse is what enables muscle contraction, transforming chemical energy into mechanical force. Today we are going to explore the fascinating world of myosin, uncovering its structure, its role in muscle contraction, and why this protein is so crucial for our movement and survival. Hello science enthusiasts, welcome back to SWA, your go-to channel for all things molecular biology, biophysics, biosensors, and radiation. I am Ayub Basiri, and in today's video, we'll dive into the dynamic role of myosin in muscle contraction. So whether you are a biology buff or fitness fantastic, or simply curious about how your body works, stay tuned to learn all about this incredible molecular motor. Let's start with a bit of history. This journey to understand muscle contraction began in the 19th century in 1864. German scientist Wilhelm Cohen, if I pronounce his name correctly, first coined the term myosin from the Greek word myos, meaning muscle. However, it wasn't until the 1940s that the full importance of myosin in muscle function was realized. Biochemists Albert Z. Grigori and Bruno Franz Straub, if I correctly pronounce their name, made a breakthrough by demonstrating that myosin, in combination with another protein called actin, was essential for muscle contraction. By 1950, scientists had proposed the sliding filament theory, which provided a model for how myosin and actin interact to produce muscle contraction. This discovery laid the foundation for modern muscle physiology and molecular biology. Now let's break down how myosin actually works. Myosin is a motor protein found in muscle fibers. It's responsible for converting chemical energy stored in adenosine triphosphate or ATP, which I make video for it, into chemical energy which generate force and movement. Structurally, myosin molecule consists of two main parts: a globular head region and a long tail region. The head of myosin molecule contain ATP's activity, which allow it to hydrolyze ATP and generate energy. It's basically a protein that converts ATP to ADP and release a phosphate. Myosin works closely with another protein called actin, which forms thin filaments in muscle fibers. Muscle contraction occurs when the myosin heads bind to a specific site on the actin filaments, forming cross bridges. Once bonding, the myosin head undergo a conformational change, pulling the actin filament toward the center of sarcosome, the basic unit of muscle fibers. This process, known as the power stroke, is represented multiple times during a single contraction, causing the muscle to shorten and generate force. ATP is crucial in this cycle because it provides the energy needed for myosin to release actin and reset for another cycle of binding and pulling. Without ATP, muscles would remain in contracted state, a phenomenon known as rigor mortis, which occurs after death. The discovery of myosin's role in muscle contraction was a gradual process involving many scientific breakthroughs. In the 1940s, Albert Z. Grigori and his colleague at the University of Szeged, Hungary, isolating myosin from muscle tissue and discovered its ATPase activity. They showed that myosin could split ATP, releasing energy that could potentially drive a muscle contraction. In the 1950s, Huxley and Niedergerk in Cambridge and Huxley and Hanson in London independently proposed the sliding filament theory, which explained how myosin and actin interact to produce muscle contraction. They used electron microscopy to observe muscle fibers and discovered that the filaments of myosin and actin slide past each other during contraction without changing length. Actually, this is exactly like this. Myosin and fibers are like that. By sliding, they go into each other, which cause contraction. These discoveries were groundbreaking because they linked the biochemical properties of myosin and actin to their mechanical role in muscle function. Over the following decades, advancements in techniques like X-ray crystallography, electron microscopy, and spectroscopy have provided a more detailed picture of myosin structure and function, allowing scientists to visualize the protein at atomic resolution. And of course, I have to mention that AFM, or atomic force microscopy, was extremely important role. AFM is a technique that we use something like needle and slide it on some surfaces. This movement and going up and down of this needle give us information about the surface, which very informative, and it could help us to understand how muscle contract. Understanding the role of myosin in muscle contraction has profound implication across multiple fields. In medicine, insights into myosin function have led to better treatment for muscle disorders. For example, mutation in myosin genes are associated with disease like hypertrophic cardiomyopathy, a condition that affects the heart muscle. Research into myosin and also contributed to therapies for muscular dystrophy and other muscle wasting disease. In sports science, knowledge of how myosin and actin interact can improve athletes' training and performance. By understanding the molecular basis of muscle function, trainers can design programs and optimize muscle growth, strength, and endurance. If I want to mention something, by molecularly analyzing the interaction of myosin and beet juice, actually red beet juice, could understand that if we give athletes beet juice before the program, or if I want to contribute in something like Olympics or something else, it actually could boost their energy and stamina. In biotechnology, engineered myosin protein are used in biosensors and nanotechnology application. Myosin's ability to convert chemical energy into mechanical work has inspired the design of molecular motor and machines that mimic biological process, with potential use in drug delivery, diagnosis, and micro robotics. Now let's delve deeper into the advanced aspect of myosin. There are multiple types of myosin, each with a specific function. The most studied type is myosin 2, which is found in skeletal, cardiac, and stomach muscle tissue. Myosin 2 is responsible for the contractile force in this muscle type and is characterized by its two-headed structure that interact with actin filaments. But myosin isn't limited to muscle. There are over 30 different myosin genes in human, and they are found in various tissue playing role beyond muscle contraction. For example, myosin 1 is involved in intracellular transport, moving vesicles and organelles within cell. Myosin V is known for its role in cargo transport along actin filament in neurons and other cells, acting like a tiny molecular courier delivering vital materials to a specific cellular destination. This movie actually showed that how myosin V actually transfer dopamine molecule across the cell. The study of myosin also interact with physics, particularly in understanding how molecular motor works. Researchers use techniques like single molecule spectroscopy to observe myosin at work, studying how these proteins convert chemical energy into mechanical work with incredible efficiency. The principle of myosin function have inspired synthetic biology, where researchers attempt to create artificial motors and machines that mimic the properties of these biological marvels. Additionally, recent studies have focused on the regulation of myosin activity by various factors, including calcium ions, regulatory proteins like troponin and tropomyosin, and post-translational modification. These regulatory mechanisms are crucial for fine-tuning muscle contraction and ensure proper muscle function, and they offer potential therapeutic target for muscle-related disease. To sum up, myosin is not just another protein, it's molecular motor at the heart of every movement we make, from the microscopic sliding of filaments in our muscle fibers to the macroscopic feat of human strength and agility. Myosin play a central role in transforming energy into motion. Understanding myosin's function helps us appreciate the complexity of life at the molecular level and opens up new avenues for researchers and innovations in medicine, sports science, and technology. I hope you enjoyed the exploration of the role of myosin in muscle contraction. If you have any question through or topic you would like me to cover in the future videos, please leave a comment below. And don't forget to like, share, and subscribe to SWA for more exciting dives into science behind the phenomena we often take for granted. Your engagement helps us to bring more of the content you love. Until next time, stay curious and help pushing the limits of what you know.